All right, it's my pleasure to introduce our first speaker of today, Jonah Nguyen. He's a graduate student at Mississippi State University working with Dr. Graham Roser and Matt Griffin. His primary interest is fish parasites such as Dermocystidium, dinoflagellates, and other understudied parasites of freshwater and marine fishes. He requested if you do encounter Dermocystidium or dinoflagellates or other obscure fish parasites that you need help identifying, don't be shy and reach out to him for that assistance. So take it away, Jonah.
All right. So this talk is basically just going to be about how to differentiate Dermocystidium species molecularly and morphologically. I kind of got into Dermocystidium last year when we were sampling red spot darters, and we got a species. I sequenced 18S of it, and it ended up being very, very close molecularly to a species found in Chinese perch in China. So then you go down the rabbit hole of, well, how do I distinguish these? How do I know it's not just that one from China? So general, what is Dermocystidium? It's a genus of cosmopolitan mesomyzetozoan parasites of fish. Other mesomyzetozoans, things like Spherothecum, Rhinosporidium, kind of in between animals and fungi. The cysts develop in the skin, muscle, gills, etc. Kind of depends on the fish host and the species of Dermocystidium. The life cycle has been shown to be direct in lab transmission studies, but the intricacies of the life cycle need more work. They're relatively host specific is what the literature has all said, but that needs more research too, of course. And it's associated with fish mortality, especially in aquaculture, but and particularly when it impacts the gills, as you'd expect. It's synonymous with Dermotheca, which is a taxonomy revision of a proposed name by Bortero et al. In 2018. There's been some adoption to that, so you might see that, but there's more validation needed for that. So how do you know it's Dermocystidium, or how do you know you have it? Well, the cyst presentation is pretty variable. This is what it looks like. Let me get out a little laser pointer here. So for example, like small round cysts, elongated cysts like these, almost like quote unquote like worm-like within like a clear bubble, commonly seen in tetras. And then in koi and carp, you'll see like raised cysts like this. And inside of the raised cysts, there's a bunch of almost like thread-like hyphae. So interesting different presentations. And as you can probably tell, there would especially be more mortality in a case like this when the gills are just full of cysts. So that kind of makes sense there. So my example and how I got into it again is red spot darters in Mississippi. So our case is a little bit different in that you can't see the cysts externally unless it was a really bad case. But most of them, you had to peel the skin off this beautiful darter and see these tiny, tiny little elongated cysts in there. So once you extract them off the fish, they're about 2 millimeters by 0.5 millimeter for this example of Dermocystidium. This cyst shape is pretty common though, an elongated type of oval type-ish shape. So that is pretty common. You popped it on a slide and you get a wet mount on 40X. It'll look about like this. You'll see it's basically just a bunch of spores packed within the cyst there. And then if you go real high magnification, you'll see something like this. So this, the spore diameter, about 4 to 15 microns, give or take a little bit. And you'll see a prominent refractile body taking up most of the space within the cell. And then these small circular granules that if you focus up or down, you'll see more of. That's if you have DIC or something similar where you could see it really well. So these types of measurements are used to characterize species, the spore diameter and their fractile body diameter. So just to do some histo, and of course, I'm not a pathologist, so I'll just tell you what the pathologist told me, but you can see the cyst here, densely packed with a bunch of spores. Not too much inflammation stuff going on in our case with our fish when intact, when the cyst is intact. But once the spores get out of the cyst, there's inflammation associated with that in our case, which of course is never good. But if you do GMS stain, which isn't super popular for dermocystidium, but you'd probably get similar results to what I got, where the spore is stained black, as you can see here, and then the cyst wall remains unstained black. So if you have something that is morphologically consistent with what I've shown, you have dermocystidium. At least you most likely have dermocystidium. It's pretty unique looking, but you never know what else is out there. So I got to hedge a little bit there. If you want to be absolutely certain, you can sequence 18S with mesomyzetozoa specific primers. Like these primers here are pretty specific for mesomyzetozoa, so pretty good for not amplifying fish contamination. So I did say that this was a differentiating dermocystidium species presentation though, so now we got to get to the more difficult part about identifying them to species. But really, this should really be called like, wow, this whole genus is a mess that someone needs to fix, because it really is kind of like a dumpster fire. I'm going to try my best to make this simple, but half of these species were described like 80 years ago, don't have any DNA molecular data available, and a lot are probably synonymous. So, and there's a lot that still need to be described probably. So there's a lot of work to be done. So first, like, why differentiate species at all though? Well, you don't know a lot of things if you can't differentiate species. So to figure out if some species are more pathogenic than others, just how narrow the host specificity truly is, which is important because if you want to know if X dermocystidium species in your fish will spread to your other fish, then we need to know what these different species are, or if there are more or fewer species described, obviously, and the geographic distribution. So are there possibly invasive or emerging dermocystidium species? Like if mine truly is the same species as the one that's in China infecting Chinese perch, what are the implications of that? So why not just use morphology to distinguish species? Well, we do, but there are not many things to measure. As I said, there's really two things that are measured. Different species overlap at these metrics as well, and measurements vary within a single species. So different cysts from a single fish can yield different measurements. So these are the same images. These images right here are of the same, at the same mag, same fish, same dermocystidium species, just different cysts. And as you can see, the picture on the right clearly has bigger refractile body diameter and spore diameter on average than the one on the left. So just to kind of hit that home, here's a graph showing spore diameter ranges for species that have molecular data. The lines show averages, these lines over here. So notice that not all species have averages available, for one, and also notice that there are multiple lines up here for my species. That's because those are the averages I got from the different cysts of my dermocystidium species. This is what the averages would have been if I would have sampled from single cysts. So not only is there significant overlap in the ranges of these spore diameters, but most species descriptions did not say that they sampled from multiple cysts or from many from each cyst or whatnot. So even if this was pretty reliable, how reliable would it even be? How are these comparisons, how good would they even be if most of these species measurements may not be representative? If, you know, for example, the decoy was from a single cyst or whatnot. So there's a lot of work to be done there, and it's a big downside of using morphology for these, at least morphology of the spores. And then the same thing for refractile body diameter. It's basically the same scenario. There's a lot of overlap between these different species here. And the averages for mine are all over the place depending on what cyst I got it from. And I would like to point out that D. cynopersi is the one I was referencing from China, that mine is very similar to at 18S. So there's some overlap there for both of these. For the spore diameter as well, there's some overlap, but it's not huge. But again, how reliable really is all this anyway with the given downsides I mentioned? So what about cyst morphology then? It is consistent within a species generally and can vary a lot between species, as you saw with one of the first images I showed with all the different cyst morphologies. So that's a good thing. But cyst shape and size overlaps a lot between species sometimes. So it isn't a unique enough of a characteristic ID to species based on it alone every single time. it's more of like a small piece of evidence you could use to help differentiate or describe a new species, but it alone is not enough, considering like 3 or 4 different Dermocystidium species, if not more, are just like elongated ovals, you know, about the same size too. So it's not unique enough of a character to for sure use it every single time. So at this point, you're like, I probably should just throw all mine away, my Dermocystidium, I don't really care about it. But I mean, try not to overcomplicate it. And I'm trying not to do that. So if fish host, infection site, and cyst morphology are consistent with a single described species of Dermocystidium, it's probably just that one. Like if you have small round cysts about 1 millimeter in size, predominantly on the gills, but maybe on the skin too a little bit, on a salmonid, it's probably D. salmonis, as you can see here. Same example used before. It's probably just that. Now, I said if a single described species, if it's consistent with a single described species, if it's consistent with more than one species, then you might have some issues. So like you can see here, there's some decoy on a common carp, and you can see the hyphae. That's what all these white little almost web-like things within this red cyst are. This is probably decoy, but the issues are you'll see in a second. So don't actually try and read this really, but this is what you might use to try and figure out what species you have. For this was published in 2021 by Liu et al. And one, it's already out of date. These species names have changed, but beyond that, these arrowed ones probably are just synonymous with decoy because they're from the same fish host, Cyprinus carpio, from the same infection site, etc. So my main point is that you can look at a table like this and get an idea of if there's a described species from that host fish yet, or from a very closely related fish host. And if there is, you can just dig into the literature a little and get a decent idea of that species and things like that. So again, try not to overcomplicate it. Hopefully you have a relatively easy ID like D. salmonis. That's relatively easy. There's still issues with it, or at least kind of easy like decoy. But if it's complicated like my case, I'm sorry. So again, my case, it's a very new host record for Dermocystidium, none reported from Ethiostoma at all. But it's the Dermocystidium species I have from them are very closely genetically related to D. cynopersi, as I've mentioned, which infects Chinese perch in China. So just trying to solve it a little bit. Sequence 18S. 18S is all that has been sequenced from Dermocystidium so far. So you can't use like COX-1 or something to try and, at least not yet, to try and differentiate species. So then I just got all the publicly available sequence data for Dermocystidium, aligned them and trimmed them to equal length, which resulted in 1,000 base pair alignment. And then I was able to assess percent identity with my Dermocystidium species. So it might be thinking now like 18S man, he comes, saves the day, it's amazing. Well, no, this is kind of like when you wish Superman wouldn't have showed up because he just ends up destroying the entire city that he was trying to save. It's kind of like that. So this is the percent table, percent similarity table. So these are all the species that have all the described species, and I threw in the cardinal tetra one in here. You'll see why I did that in a second. But this shows as a whole that 18S is really not great for identifying these because there's 4 species within 0.5% to mine. They're just very close together. There's not a lot of diversity happening at 18S with this genus. Besides for a D. percy, that's just weird. That's it's low percent. I don't know what that's about, but imagine also that it would be even more crowded in this table if more species had sequencing data. So you can just imagine. So now you see D. cynopersi, that's the one in Chinese perch I keep talking about, almost close, or is 100%. It depends on the isolate to mine. So you might be thinking, well, I mean, Jonah, don't kid yourself. It's probably just that. And it might be, but there's really a razor thin margin of error here. Because Langenmeyer et al. In 2014, which they did good work for their time in 2014, but now we know more and there's some issues. So they identified what they call D. salmonis, which infects salmonids from cardinal tetra. And they called it that because it had a 99.7% similarity to D. salmonis. So they inserted their GenBank accession. And so now in GenBank, there is an accession for Dermocystidium salmonis that is from a cardinal tetra. But in 2021, that same Langenmeyer isolate was found to be basically 100% similar to D. angeli, which infects American eel. So that proves one of two things. Either it's very non-host specific, which goes against what the literature has said, or 18S sequencing is just very inadequate, which is common for a lot of taxa. So you can see a cardinal tetra with Thermocystidium, the characteristic little clear bubble with little worm, quote unquote, on the inside. There's a reason why I think it's more of the second one, because one, like I said, the literature has consistently noted that it's relatively host specific, only infecting related species. So for example, in a fish tank with a cardinal tetra, these other fish species, I've read quite a few anecdotes from people, aquarists keeping fish. These don't get Dermocystidium, at least not visibly, maybe histopathologically, but there's no visible signs of it. Other tetras, like emperor tetras or neon tetras, those might get it from the cardinal tetra, but these wouldn't. So that leads me more to this conclusion. So if 18S is so inadequate for Dermocystidium that two species are basically 100% identical at 18S, then of course D. cynopersi could certainly be a different species than mine. It could be. So what am I going to do? What have I been doing to try to fix this? So we can try to sequence some new DNA markers from Dermocystidium, especially mitochondrial DNA markers like COX-1, to see if those are better at differentiating Dermocystidium species than 18S's. So next generation sequencing is called that for a reason, apparently, because it's pretty cool and pretty good. So DNA extractions from the Dermocystidium from red spot darters were sent for Illumina sequencing rather at Minnesota. And that got me about 5 million 300 base pair reads, which is pretty cool. And from that data, I was able to design primers and sequence with Sanger, Site B, COX-1, and 28S. And to help with this project, I also got some decoy sent to me. So here's that decoy specimen. This isn't actually from, that wasn't actually what was sent to me. That's just an example image, but this is the cyst that was sent to me. And you can see how small these hyphae are. That's what all these little thread-like things are. These are 1 millimeter measurements in this ruler here, and this is the red cyst. So Dr. Jesse Sanders sent me a decoy specimen. And from that, I have preliminary 28S data, but I'm still waiting on Illumina data to sequence more markers such as COX-1 from that. But with that, I'm able to make a table like this. So you can see that going this way to the left, there's a decreasing percent similarity to my Dermocystidium species. That's what this is showing. These are showing percent similarities to my Dermocystidium species. So you can see decoy at 18S, 99.4% similar to mine. At 28S, only 90. So we're already seeing a better, lower percentage there. That's really promising. This is the only other Dermocystidium species I have, so that's all I have from that. But there's other related organisms, such as these other mesomyzetozoans here, that show the same trend, where COX-1 and Site B have way lower percent similarities, such as 67% versus 94% for 18S. That's a lot better. So it essentially shows that there are better markers in 18S, which we kind of knew because other taxa, some other taxa have the same thing. So next step for me, I need more Dermocystidium species. That's why I always solicit for it. So if you find it or you think you found it, you can just contact me, let me know. But getting more species would allow me to confirm which markers best differentiate Dermocystidium species. I can also, once I get more of them, design general Dermocystidium primers so that basically the goal is achieved then. Researchers all over the place can accurately ID to species by using these generic Dermocystidium primers. But to do that, obviously, I need more Dermocystidium species to design these generic Dermocystidium primers for these new markers. And then summary for you is identifying the genus level is relatively easy, even just morphologically, but identifying the species is more difficult. But try to keep it simple where you can. If everything is consistent with the Dermocystidium species, then it's probably that one. If you have a more complicated case with a high 18S% similarity to already known and described species, then you gotta kind of just hang in there a little bit, be speculative about what you say and whatnot, but hang in there. It might be a new species, maybe not, but we need new DNA markers just to be sure. So that is all. If you have any dermocystidium questions, you can email me. But that's it. Yeah. Thank you all for listening and watching.
Great. Thank you very much, Jonah. Excellent presentation and decomplicated it for me. And hopefully somebody out there has some dermo to send you. You've got some time for questions if folks have them.
Hey, Jonah, great job, man. Real quick, and maybe I missed it. I should know this, but I don't. What is the cyst morphology of like your isolate compared to the
So
the Chinese perch? Is it
is
it in the same way, or is it like?
I gotta go right back here. I left that out because I didn't think I'd have enough time, but it's pretty similar. They also basically look like these little ovals like this, maybe a little bit bigger, but they basically look like that. But they're visible from one. They also infect the fins. I haven't seen it on the fins yet. Doesn't mean it's not possible, but I haven't seen any on my darter fins. It also has been seen in the gills for them. I haven't seen it in the gills, but the cyst morphology, in that it's just an elongated oval, is basically the same. Yeah.
Just out of curiosity, now that I got you on the line, how many described species are there?
There's 19, I think, is what I had in that table. There's 19. Yeah, there's definitely, well, I'm gonna hedge. There's probably more than that, but there's 19 so far. And again, a lot of those are probably synonymous of the ones that are described, but a lot of work to do.
Seems like there would be more than that out there.
Probably. I don't know if there's any messages in the chat too. I don't have the chat pulled up.
Yeah, nothing in the chat, but
Okay. All right.
well, good. Thank you very much, Jonah. Excellent
Thank
presentation.
you.